Hi everyone, this is your biology class. I'm your teacher Seema Atreja. Student, in this video we are going to discuss how selectable marker help in selection of transformant. In previous video we studied the structure of vector PBR 322. We also study there two selectable marker. One is antibiotic resistance gene and the another is enzyme producing gene. We consider both these markers here and also discuss how these markers will be able to identify our transformant. So these are our two marker gene. One is antibiotic resistance gene, and in case of PBR 322 vector, there are two antibiotic resistance genes are present. One is ampicillin resistance gene and another is tetracycline resistance gene. Second marker gene is the enzyme producing gene. Both these marker gene helps in selection of the transformant. This is our vector, that is plasmid, act as a vector. It consists of two antibiotic resistance gene. One is tetracycline resistance gene and another is ampicillin resistance gene. At the tetracycline resistance gene, there is a site that is BAM-H1 site. Along with this, restriction endonuclease enzyme is present with us. Desired gene which we want to insert in this vector and construct recombinant DNA or recombinant plasmid. For this, also present with us is the ligase enzyme. First of all, this restriction endonuclease enzyme cut this vector at specific site, which is the BAM-H1 site. It recognizes the specific sequence to cut on the vector, and that specific sequence are present at the BAM-H1 site of tetracycline resistance gene. So this restriction endonuclease enzyme cut this plasmid at BAM-H1 site. After cutting this, ligase work here. What is the function of ligase? It help in ligating these desired gene at BAM-H1 site. So these desired gene are inserted into the vector at BAM-H1 site of tetracycline resistance gene. And this becomes our recombinant DNA, or we can call it as recombinant plasmid. In the next step, we insert this recombinant plasmid or recombinant DNA into the host cell. This is our host cell inside which we put this recombinant DNA. Now, as the host cell multiply, this recombinant DNA also multiply, or growth of the host cell takes place. Now we put this host cell, which consists of this recombinant DNA, into a culture medium where ampicillin antibiotic is present. When we insert this recombinant DNA into host cell, it becomes transformant. What do you mean by transformant? Which consists of recombinant DNA or vector in which desired gene are present. So host cell become transformant because inside this desired gene is present. When we put this transformant into ampicillin antibiotic containing culture medium, it will grow because inside it ampicillin resistance gene is present. So in presence of ampicillin antibiotic, it will grow because ampicillin resistance genes make it possible to grow in presence of ampicillin antibiotic. Now we transfer this transformant cell into next culture medium where tetracycline antibiotic is present. Now this transformant do not grow in this culture medium where tetracycline antibiotic is present because desired gene are inserted at tetracycline resistance gene. So tetracycline resistance gene here becomes inactive. So this transformant do not survive in presence of tetracycline antibiotic. When we transform this transformant into ampicillin containing culture medium, this will survive and growth takes place because ampicillin resistance gene are present into the transformant. But when we transfer this transformant into culture medium containing tetracycline antibiotic, it will not grow. Growth does not take place because desired gene are inserted at BAM-H1 site of tetracycline resistance gene. So tetracycline resistance gene becomes inactive. So in presence of tetracycline antibiotic, it will not grow. So this is our transformant. What do you mean by non-transformant? In which desired gene is not present. So then this non-transformant grow in both culture medium. If the culture medium containing ampicillin, it will grow, and if the culture medium containing tetracycline antibiotic, it will grow. This is known as the non-transformant because desired gene are not present. So both type of resistance gene, ampicillin resistance gene and tetracycline resistance genes are working in case of non-transformant. So growth of non-transformant takes place in both type of culture medium, whether it contains ampicillin antibiotic or tetracycline antibiotic. So in this case, what we are observing here, one antibiotic resistance gene, that is ampicillin, helps in selecting the transformant, whereas the other antibiotic resistance gene, that is tetracycline, becomes inactivated due to insertion of desired gene at BAM-H1 site of tetracycline resistance gene. So thus it helps in selection of transformant in which desired gene are present. So antibiotic resistance gene is the marker gene which helps in the selection of transformant. Now we discuss the another marker gene, that is enzyme producing gene. How enzyme producing gene helps in the selection of transformant. This is the second case. Enzyme producing gene, for example, mostly we take marker gene as LAC-Z gene, which is responsible for the formation of beta galactosidase enzyme. This beta galactosidase enzyme is responsible for conversion of a substance X-gal into a product. What is X-gal? It is a white color substance. It is a modified version of galactose. X-gal is a white colored substance. It is a modified version of galactose. This galactose, in presence of beta galactosidase enzyme, is converted into a product which gives blue color. This is what we will be using in selection of our transformant. This is our plasmid, and in which act as a vector, and in this vector or plasmid there is present LAC-Z gene. which is responsible for the formation of beta galactosidase enzyme, which is needed for the conversion of X-gal substance, which is white in color, and after conversion it converted into a product which gives blue color. But now what happens? In this plasmid we insert this desired gene at the position where LAC-JAD gene are present. When these desired gene are inserted at the LAC-JAD gene site, it becomes recombinant DNA. Due to insertion of desired gene at the position where LAC-JAD gene are present, the LAC-JAD gene become inactivated. This process is known as insertional inactivation, and we also called it as blue-white conversion. Why this is called insertional inactivation? By insertion of the desired gene, the main gene LAC-JAD becomes inactive. Because of this, when we insert this recombinant DNA into host cell, the gal substance do not convert into blue product and it remains white. Insertional inactivation is very important because blue color do not appear. It means it becomes transformant. It consists of the desired gene. This is all about in this video. Thank you so much.